Hello everyone! In this video, I'm going to give you an overview of the services we can perform at Applied Pathology Systems within our laboratory. We are going to cover histology, pathology, as well as some of our special capabilities. As a histopathology laboratory, we provide comprehensive histology services to support different types of projects. Our first general category of services is our histology services. Here, we can process fixed tissues, cell lines, fresh tissues, and we can process any paraffin slide blocks that you submit. Over the past several years, we have developed a lot of special histology capabilities. For example, we are able to process special tissues such as mouse brain, eyes, or bone marrow. We can construct tissue microarrays using the tissues you provide, and we can process organoids or any other types of tiny cell cultures. Lastly, we can also support apoptotic studies by performing tunnel assays. We perform many types of immunohistochemistry stains, including antibody validation and staining, antibody screening, therapeutic lead antibody characterization, and multiplex immunofluorescence staining. When these slides are completed and stained, we can either take digital images or scan the slides for you. We are able to scan both bright field and fluorescence multiplex standard slides. Our second general category of services is our pathology evaluations. All of these evaluations are performed by our in-house board-certified pathologists that are capable of evaluating different diseases or disease models across different types of tissues or organs. When the evaluation is finished, we can provide a report if necessary. You can then use this report to support any of your submissions or publications. Our next category of services is image analysis. We can perform cell segmentation, pixel quantification, spatial analysis, or object measurement. With all of these capabilities, our team of scientists is able to support clinical trials or clinical studies. Next, it's important to mention that APS just recently became CLIA certified, which means that we're fully able to provide anatomic pathology clinical services for patients in clinics or hospitals. Lastly, we have a list of human specimens that allow us to support your clinical-related research. This next slide contains pictures of our wonderful laboratory. We are located in Shrewsbury, Massachusetts, which is less than an hour drive from the Boston-Cambridge area. We are fortunate enough to be situated very close to some of our clients and laboratories that are local to the area. Now, I would like to go over some of the technology and equipment we routinely utilize in the laboratory. Along with the regular, well-known lab equipment, we have all the key histology equipment that allows us to perform different types of histology projects. We have an automatic tissue processor, a tissue embedding center, microtomes, cryostats, different types of autostainers, cover slippers, as well as several imaging systems. Within our imaging systems, we have a fluorescence microscope that can take up to 5 channels in your IF studies, but it has the capabilities of even more channels if necessary. Next, I want to talk about the whole slide scan system that we have within our laboratory. It's different from the traditional way of taking images that involves a camera and microscope in that it can take many images that cover all areas of the tissue found on that slide, and then its own software will automatically stitch all those images together to form one condensed image of the tissue. This image can be shared very easily with anyone through computers. Now, this system is better than regular imaging because it's an unbiased whole view. It's fast, and you don't have to wait until the slides are shipped to you to see the results and make your analysis. It's very convenient to view, edit, and show these images to your collaborators and colleagues. But I think the most important feature of our whole slide scanner is that all of the files are permanent electronic files, so you don't need to worry about a slide going missing, a slide getting broken, and you don't need to worry about the decay that comes with immunofluorescence staining over time. In-house, we have this machine known as the PhenoCycler Fusion System that we purchased from Akoya Biosciences. This system uses a combination of 2 distinct units, the high-quality fluorescence scanner, which is in the middle, and then the automatic IF stainer to the left. With this system, we can easily stain many markers on the same slides, and theoretically, we can stain 100 markers on the same tissue. The scanner itself has a very narrow wavelength and can produce extremely sharp and high-quality images. Next, I want to go over what sets APS apart from many other laboratories. Firstly, our team members are very intelligent, eager to learn, and are always one step ahead of knowing what is to be done in the lab. Our laboratory personnel includes board-certified pathologists, histologists, medical doctors, scientists, IHC specialists, and image technicians. Our team is comprised of people who work together to make sure that we understand the science, so we are able to deliver good quality data to our clients. In our laboratory, we have specific SOPs for each stain to ensure quality and consistency. Here on the left is an image of a lower magnification H&E stain of a mouse lung with inflammation of foci, and on the right, we have a higher magnification of the slide that shows the details of the foci, which shows how it's filled with neutrophils. At APS, our overall mission is to provide our clients with high-quality services with a quick turnaround time and provide the flexibility and convenience for them to submit samples and submit their requests. Working with a scientist at APS, our clients are able to voice their needs and submit all of their requests together, and in turn, they can make their study more cost-effective. In the end, APS strives to support our clients' research and aid in combating disease.